All right. Thank you, everybody, for joining us today. I'm really excited to be here with Warren Huff, who's the former CEO and chairman of Reata Pharmaceuticals, which was acquired by Biogen in 2023. And he founded Reata in 2002 and led the company for that entire 21-year journey, which had lots of ups and downs, but culminated in a major up, which was the approval of Skyclarys, which was the first FDA-approved therapy for Friedreich's ataxia. And along the way, they did some incredibly pioneering work with using natural history studies and really innovative trial designs to take a challenging rare disease population and actually get it over the finish line, which is a major win for patients and other stakeholders. So, Warren, thank you for joining us and excited to dive into your story and the topic a little bit more today.
Patrick, thanks for having me. I'm really pleased to be here.
I would love to just start a little bit with the origin story of of the Skyclarys drug, which is an amazing story in and of itself. If you could give people just brief background if they're not familiar with the story there, and in particular some of the innovative points that I touched on around how you used natural history study data and partnered closely with patient organizations, I think would be really helpful to just anchor people on what we're talking about here and how they might apply it to their own work.
Sure, I'd be happy to. So Skyclarys is, one of a new class of drugs that doctor called as kind of yin and yang or directly counter-regulatory to NF-kappa B. And so, it's a key regulator, of the inflammatory response. And it's known now that it's also a key regulator of mitochondrial function, that those work together. And so it basically restores mitochondrial function, when it's been impaired in an inflammatory response. We worked all this biology out for years, and we used the drugs in multiple indications. We had a separate drug, bardoxolone, that we tried to get approved in CKD. We weren't successful there. But we had an independent program in Friedreich's ataxia, a rare neurodegenerative disease. It's actually the most life-shortening neurodegenerative disease. It's like ALS for kids, a terrible disease. And it had been a wasteland of development. There had been like 15 failed programs. It was very tough. But, and I'm sure we'll talk about more, the patient group had made a lot of groundwork, to develop drugs in the space, including a natural history study. But they had a clinical research network. They'd done basic work showing NRF2 was critical, to the disease and was suppressed. They actually approached us to use our drugs for this application. And we ultimately agreed because the setup was so good, and they worked out endpoints, with the FDA. So we did a development program in FA, which culminated in a successful randomized controlled trial of a year in duration. It's very conventional design because there was no approved therapy for these patients. So it was placebo and active drug, Skyclarys, for a year, measuring their neurologic function with the approved endpoint, the Friedreich's ataxia rating scale, which was kind of a standard neurologic exam adapted to this disease. And we showed very positive results. There was actually a recovery of function initially in patients on active, so a one-time improvement in their neurologic function, and then that was maintained basically for the year, while the placebos had a placebo effect, which they then lost and then declined throughout the year. And so we had an improvement in the mFARS of a few points, which was meaningful. It's 1 to 2 years of progression. And it was the p-value was a 0.014, p-value, which is very good for a rare disease. This is a very rare disease, by the way. And so we went to the FDA with that, and there was a lot of back and forth. But a critical issue was it was a single study approval in the rare disease space. And I know this has been a dynamic area recently among sponsors and regulators. How do you put a clinical package together in this very difficult space with very few patients and produce enough data basically to support, to provide the agency, with what they need for approval? Ultimately, we got the drug approval on a basis of that study, which was very solid, very internally consistent, solid. It didn't quite meet the standard for high statistical significance and a single study approval because the p-value was above 0.01, but we were
Mhm.
right on the threshold of it. And so we ended up supplementing the NDA with an externally controlled study using natural history data, and that convinced the FDA. We got approval in February the 28th, 23, launched the drug, I think 6 or 8 weeks later in the US, and we're working on our European approvals when we basically got approached by two large pharmaceutical companies, which ended up putting us in play. And we sold the company to Biogen for 7.3 billion, and they have the drug Skyclarys now, and they're doing an excellent job with it.
Yeah. Yeah, that's an amazing story, and I think it has landed in great hands. They seem to be doing a really great job with it. I'd love to come back to the that supplementary data you provided and the natural history, essentially external controlled study that
Mhm.
you talked about. How important was that? It sounds like it was pretty critical. Was that obvious to you from the beginning of engaging with the patient organization that this was going to be a critical piece of data, or did it kind of the pieces fell into place almost towards the end, and you were very glad that they'd done the decades of hard work they did to collect the data so scrupulously.
Yeah, at the time, so to give a little of background, at the time, the FDA and particularly the Neuro Division were definitely struggling with multiple projects, multiple sponsors that had single studies that were positive in the rare disease space, and then how to apply what's called FDAMA 115. And so in the US, there's a couple of ways to get approval on a single study, which is again critical in the rare disease space. And one is to do two studies, but actually the conventional is two studies with a 0.05 p-value, really not feasible in the rare disease space, or a single study that's highly statistically persuasive. Again, very difficult because you can't produce the numbers to drive your stats. And so Congress passed a thing called FDAMA 115, which basically said you can get a single study approval that's not highly persuasive statistically if you have confirmatory evidence. And the FDA has been working with guidance documents and everything to try to figure out where to land on what's confirmatory evidence. And it can be things like if you're doing enzyme replacement, the mechanism,
Yeah.
is confirmatory. And there's several ways to do it. But one is that you can use natural history data to support the data from your RCT, your randomized controlled trial. And we went further than that. We did a design that was effectively an externally controlled. If we pre-specified it, it would have been an externally controlled study. So throughout our process of review of our data, we had this ongoing dialogue with the FDA, and other sponsors were having this as well about how they were going to apply FDAMA 115. And there were many different approaches that could be taken. Could be you could use that data, just comparing it to your progression rates. You could do what we did. And there were also some discussion of using things like a delayed start analysis, where you basically you roll your patients over to your long-term study, and you look at the difference in the rates after another year or two in the ones that had a delayed start because they were on placebo. So all of this was in a dialogue with them, but it came together about midway through our review process. We had 3 years of data in an open label study that we rolled all the patients over to. This is a critical point for sponsors. And FDA liked the idea of, because it was very rigorous, of using the natural history data for an external control.
And the key thing that you were aiming to show or that you did show with the natural history data was it around rate of progression in the absence of any intervention? Was there anything about certain subtypes of patients that respond differently? Maybe you can talk a little bit about the
Yeah,
things you were
if
looking to
you
confirm.
yeah, if you'd like, I could talk about the design of that and kind of
Yeah.
how it related to our randomized control trial. So it's all very simple. We did the RCT at 1 year. And when the patients finished, we rolled them over to an open label extension. They all got Skyclarys, and we tracked their Friedreich's Ataxia Rating Scale, the primary endpoint, scores and other things along the way as they stayed on. And the placebos rolled over as well. And critically for sponsors and for us, we did not disclose to the patients what their original treatment assignments were.
Mm.
So, and then you're accumulating data while you're going through your reviews and stuff in your open label study. With respect to the natural history data, FARA had done a wonderful natural history study. They had patients that had been on as long as 15 years, and it was run basically like a controlled trial. And so patients came in every year. They got, incredibly detailed baseline data on these patients. They then had their clinical network that really knew the endpoint and how to conduct the mFARS and everything. They were the sole sites in the study. And luckily, there were no intervening approved therapies. So we got this pure data set, of many patients seeing, year after year what their progression rates were, on the approval endpoint.
Yeah.
And the most important aspect of that is that it empirically showed what baseline characteristics drove the progression rate. And this, I think, may be one of the most valuable things about these natural history studies. First, they allow you to power your randomized controlled trial, right? But then in the regulatory context, you've got this patient set with these baseline characteristics that and their progression rates. So that allowed us to basically use them as external controls. And so the way that worked was we basically knew that the age of onset, so how early they were diagnosed, their sex, their age, their baseline mFARS, and their ambulatory status, predicted what their progression rates were going to be. And so we worked out with the FDA that we will match patients from our long-term extension with a patient from the natural history study that had very similar to identical kind of, characteristics that would drive their rate of progression. And then basically you run the match, and then you do the stats like they were a placebo. And it's a beautiful thing, really. And ours showed a dramatic effect of slowing of the rate of progression. And so related to that, we did it 6 times to show that it wasn't just a fluke, kind of like a Monte Carlo. So we did
you resample it a few times. Yeah.
And we reset it, do it again and again and again. And, and each time it showed a robust slowing of the rate of progression, of patients versus their matched control from the natural history study.
Yeah. I mean, is this different from the concept of digital twins that people are talking about? It feels like it's certainly what you've done there could be described as a digital twin type approach as well, right? Is that fair to say?
It's similar, yes.
Yeah. Yeah, I mean it's a whole area. I think you all created a pathway in some ways. This was the first or one of the first times that people have taken this approach, right? How replicable do you think this is across the 7,000 to 10,000 other rare diseases that we need to get out there and find treatments for?
I think it's very replicable. Many of these diseases, even if you have an approved therapy, as long as you got data in the natural history study on the rates of those patients. So I think it's very replicable. And one of the lessons to sponsors is plan for this up front. When you're designing your RCT, plan for the rollover, make sure you don't disclose the treatment assignments, collect the same, endpoints, and ideally work out with the FDA the study that you're going to do, the analysis that you're going to do, in the external control. So the only thing that we didn't do that would have strengthened it was to have anticipated all of this instead of the FDA when we started the RCT. Well, at 3 years after the rollover, we're going to cut the data off and we're going to do a match and we're going to use these factors
Yeah.
and we're going to run those stats. That would have been basically a controlled trial, a externally controlled trial that met all of the requirements of the ICH guidelines for a well-controlled study. And so sponsors today can put all of that in front and have a very strong package.
What can sponsors do if there isn't an existing natural history study, or maybe related to that, if what are the common deficiencies, I guess, data isn't always being collected in that kind of clockwork, really robust fashion, or what are some of the other things that you need to look out for to make sure that you can get to the end of the line and have what you need to have?
Clearly, we had an ideal situation because it was the same clinicians in the natural history study that were doing the randomized controlled trial. So there was substantial overlap, which again was really reassuring to the regulators. And so, that's a pitfall. Like if you've got a very different set of clinicians that are seeing the patients in the natural history study versus the patients that are running your placebo-controlled studies, that's an issue. You've got to collect robust baseline data. And you really need to know what drives the progression rates and the events, and have that worked out, And so if you don't, it's a problem. What's really the use of the data? And I think sometimes it's done where it's thought of as kind of an afterthought. It's not rigorous. Like in Farah's case, they had all the investigators met annually, they published data, they reviewed it all, they treated it all, they had integrity reviews of the data, by clinical research associates. They ran it like, a trial that would support an approval. And so another thing is it could be messy, because the sites change, or the endpoints. And probably one of the biggest ones are it's not big enough. Like if it's not big enough to get a good match, right, then it's really not usable. So you really need all those things.
What
So
was the size of the randomized trial, and then the size of the natural history study to help orient people on that?
I think I'm doing this wrong. I could get this wrong, but I think it was around 600 or 700 total. Some of them, they were not all usable to us. We were only able to get a 1-to-1 match. The ideal scenario is to get a 2 or 3-to-1 match. That makes it very strong. Like if it's, you get 3 patients that are close and you average those results and you compare those, it's obviously very robust. But we got good matches. Like we had 1-to-1, but all the metrics, gave you a good match. And so you can think of that like you would look at the randomization in your control trial. So, when you do your propensity matching for your your study, you look at, well, what are the characteristics of that population, the means and medians of their baseline mFARS or et cetera. And so you're trying to get those very close, just like you would get a good randomization, in a controlled trial.
Yeah. And how are these natural history studies getting funded typically? Are they going for NIH grants or are they nonprofit organizations that are doing philanthropic funding, or are sponsors kind of coming together and saying, hey, we're going to be working on this and it needs to exist? It'd be interesting to learn a little more about that.
I think now, and particularly in rare neuro, the patient groups have gotten very active following the example of Farah. And there were others doing this as well. It's best it's obviously ideal if the patient group, is essentially the sponsor. They'll form an independent, committee to run the trials and everything. And I know it's easier for them to raise money to support those studies, but there are NIH grants for them as well. And of course, there are times where as a sponsor, you have to take this on. That's difficult if you particularly have a slowly progressive disease, right? Just because of the time it takes to accumulate the data. And then what happens if you stop the program? So it's best if an outside organization does it. And I think there's a lot of interest in it now. And I think there's a lot of awareness among patients now that, this is something that they can do. They make a big commitment. They usually have to travel in these rare diseases. They usually have to travel
Yeah.
and they go for years, and there's no real hope of of a therapy, immediate hope of a therapy for them. So it's a quite they deserve a lot of accolades, the patients that volunteer for these studies.
Were the folks at FARA, were they clinical development, clinical research people? How do they, because that's a hurdle too, right? You have to know how to design a multi-year, multi-country, hundreds of person natural history study, which is a
Yeah,
unique skill in and of itself. And you can hire that in, but to have the vision and foresight is pretty exceptional.
yeah. Well, I've said this many times. They're an extraordinary organization. It's an incredible group of people. The one of the key people involved, Jen Farmer, who's the CEO of FARA, she had been in clinical research, at CHOP, and saw patients in one of the top KOLs, Dave Lynch's, practice before joining FARA. And so, yeah, she was really an asset for them in terms of, getting this design. But the other founders they were on this even before she joined. They just got advice that you need to start
Yeah.
collecting this data, and they just did it. They did whatever it took to try
Yeah,
to
it's amazing.
positive therapy. Yeah, they're an incredible group of people, and they're a great example of how big an impact a patient organization can have. Because it was the natural history study. They worked out the MFARS with the
Right.
FDA. They recruited KOLs, put a whole clinical research, group together, as well as funded basic research.
Yeah, wow. Yeah, I was going to ask, it sounded like you said before that they established the endpoint with the FDA, which is there's a series of hurdles to overcome here. And in many cases, is that probably the first hurdle to really get people organized and agree with the FDA an endpoint that you can actually go after? Because the point you made earlier, if the natural history study isn't lined up with the trial, then you're not going to be able to use it in the first place, right?
No, that's exactly right. And that went hand in hand with, recruiting a KOL group and a clinical research network, and getting consensus among them
Yep.
about FARS and then bringing FDA on. I mean, that was a long, long process, but it paid off in the end.
Yeah, absolutely. What do you see from here? So more companies, hopefully, and patient organizations are going to be collaborating around this approach. What other kinds of things do you think, based on what you've learned going through this, do you think either people are already starting to do that's interesting, or maybe they aren't doing it, but they should be because the path is clear now?
I think our approval definitely showed, gave a roadmap for how to use this kind of data, and also at least one example of how FDAMA 115 can be met, what does constitute, confirmatory evidence. So I'm aware of like many projects now that are using that example, to guide the design of their pivotal studies and then what they do, kind of as we've been discussing. Um, and and so I think that's the good news. I think, as we've kind of alluded to, what do you do if you don't have, that underlying data?
Yeah.
And I think a lot of people are struggling with that, you know.
Yeah. Is there a solution you see to that besides getting, seeding patient organizations, helping them get this started?
Well, one thing you can do, and some of this was developed by the FDA for Alzheimer's programs, and I mentioned it briefly earlier. There's a thing called a delayed start analysis. And so even if you don't have natural history data, you can roll your patients over to an open label extension. And like in our case, so you had a group of patients that were in the open label extension that had been on active drug, for a year. And then you have a group of patients that were not on active drug, the
Yeah.
delayed start group, right? So they start drug there. And then you can look at them over time. So 1, 2, 3 years and look at what happens to them. And if they converge, so if the former placebo or the delayed start patients catch up, basically, to the original active patients, right, then that tells you that you don't have a disease modifying therapy, right?
Yeah.
But if they stay separated or the separation increases, which was the case with us, then it shows you it's a strong indication, of a drug effect and a disease modifying drug effect. So
Yeah.
that can be done with you're essentially using your actives as controls, for
Yeah.
that analysis. And so that's something everybody should bake in these kinds of diseases, even if they don't have a natural history study.
Yeah.
Obviously, then you can, try to collect that data. I mean, some people, I think, try to collect that data from clinicians that treat the patients. And there could be situations where that could be persuasive, I think. And thirdly, some of them, start up
Yeah.
their own studies.
Yeah. And sometimes you have to make that multi-year commitment to figure it out, right? And that's just the
Mhm.
only option. When you think
It's
about designing those, whether it's a patient organization or a sponsor, how do you think about data quantity versus quality or density, however you want to think about it? For example, you for a fixed budget or approach, you could rigorously measure 300 patients, or you could take a much broader approach where you go and scrape medical records and pull in information and you get much messier data, but maybe you get 10 times as much people. Is there any place for that bigger, more real world evidence type approach, or do you really need something that's like an RCT but not interventional?
Well, this is my own bias, right? And of course, you can always try to work this out with the FDA in advance,
Yeah,
right? But I go for rigor. I mean, I think they go for rigor.
yeah,
I mean, I think they want to know basically that this is real, and they're legitimately skeptical. That's their job,
yeah. That's
right?
their job. Protect people by being skeptical. Yeah,
That's their job. So again, I think we had a great situation that I think they were very happy with because you had
yeah,
this long-term study. It was the approved endpoint. There was no intervening, therapy that
yeah,
confounded progression rates. We had good matches, like the things that influence the rate, of decline in the mFARS or increase in the mFARS, which was bad. Were well known. It had been generated from the data and published and peer reviewed. All of that was done. So that's the kind of thing, is going to persuade FDA.
yeah. Is there a place for these in common diseases as well, or genetic subtypes of common disease? How do you think about that?
Absolutely, right. Yes, and of course it's easier to do there,
Mm,
You have large patient groups and it's easier to do to collect really robust data sets. And by the way, another aspect of this that is important for sponsors is that this data can be really helpful with payers.
right.
Um, I think it was very helpful to us. Like you had a really good result in 1 year, but then when you saw that it was, dramatically affecting their rate of progression over 3 or 4 years, essentially an independent study,
Yeah.
that was highly persuasive to the payers. So it's not just about approval, you're also showing what the long-term benefit of your drug can be.
Do you see a lot of biotechs thinking this far out? Because it's hard in some ways to say we're going to invest in something like this to help with payers that may come many hurdles down the road. How do you like when you work with biotech companies? How do you help the CEOs or management team to think about okay, what's right in front of our face that we need to get through versus the thing that we'll thank ourselves for down the line?
Hopefully, Patrick, we're helping right now.
Yeah,
The answer is, no, it's such a difficult thing to do, to build a development stage biotech in a novel area where you're really trying to have an impact on patients. And, it's all difficult.
Yeah.
And, you've got so many challenges along the way. It's hard to think like that. We didn't. I mean, we did have these ideas, but we didn't put them into practice or work this out before we had our positive, data in the trial. I would now the second time around.
Yeah.
So it's a difficult thing, but it pays off to really think, when you're designing your randomized controlled trial about what to do with these patients, how to do it, how to maximize the data from your long-term extensions, all of that. Now there are roadmaps. And so there's no reason not to do it.
Yeah, absolutely. As we close out here, you're about a year from the acquisition, right? Give or take. What's changed for you? What are you focused on now? Just for people who are curious about what's next.
Besides my skiing,
Yeah, that's important. It's about that time of year.
yeah, well, I'm working. I wasn't ready to retire. Actually, was really excited to continue Reata and apply the technology to other diseases, particularly in the neuro space. But that's the way things go. And so I'm working with two of my longtime partners, financial partners, and we've been actively looking for projects that could have high impact on human health, and that could have help with both capital and but mainly manage building a team,
Yeah.
and strategy and everything. And so we've identified a project we're working on now, and so I'm not going to CEO anything, but I'm excited to help you know on a project that could really have a significant impact in human health. So.
Amazing. Well, thank you for sharing all these hard-earned lessons with us today. I hope people listen to this and or watch it and take something useful for them. And ultimately, we're all, especially working in rare disease, we're doing very important work to try to get therapies to patients who need them very much. So, congratulations to you and the whole team for all the progress there, and thanks for sharing your knowledge with us.
Sure, very happy to do it. I would just congratulate everybody in this field. It's really difficult, and it's a great thing to do. And so, and Patrick, thanks for having me. I really
Yeah.
enjoyed it.
Always my pleasure. What it's hard, but what a great way to spend 20 years of your life, right? Especially with this outcome.
Yeah, yeah, no, absolutely. I'll think about it on my dying breath, probably.
Definitely.
Hey, we did that.
Absolutely.